Have you ever stared at a nutrition label and wondered, how do they actually know there are 10 grams of protein in this? It seems like magic, right? Well, today we're pulling back the curtain and stepping into the food science lab to uncover the secrets behind those numbers. It's not magic, it's some seriously cool science. First step, how do they measure water content or moisture? The most common method is the oven drying method. It's pretty straightforward. A scientist takes a precise weight of the food, pops it into a special oven at a high temperature, usually around 105 degrees Celsius, for several hours. The water evaporates, and they weigh the food again. The difference between the starting weight and the final dry weight is the amount of moisture. Simple, but super important, as it affects the concentration of all other nutrients. Now for the big one, protein. For over a century, the gold standard has been the Kjeldahl method. This is a classic chemistry process. It involves digesting the food with strong acid to convert the nitrogen in the protein into ammonium sulfate. Then, through distillation and titration, scientists measure the amount of nitrogen. Since proteins are, on average, about 16% nitrogen, they can multiply the nitrogen result by a conversion factor, usually 6.25, to get the total protein content. It's a bit complex and uses some harsh chemicals, so a more modern, faster method is the Dumont method. This involves combusting the sample at a very high temperature, which releases nitrogen gas. A detector then measures the amount of gas, and from that, the protein is calculated. It's much quicker and safer. What about fat? This is where things get interesting with a technique called solvent extraction, often using an apparatus called a Soxhlet extractor. Imagine a continuous cycle where a solvent, like hexane, washes over the food sample. This solvent is great at dissolving fats, but not other components. It drips through the food, carrying the fat with it, then gets heated, evaporates, condenses, and drips through again, repeating the cycle for hours. Eventually, all the fat is collected in the solvent. The scientist then evaporates the solvent, leaving behind just the pure, extracted fat, which they can weigh. Next, we have ash. This sounds strange, but ash in nutrition refers to the total mineral content, things like calcium, potassium, and iron. To measure this, a sample is placed in a ceramic crucible and heated in a muffle furnace to incredibly high temperatures, like 550 to 600 degrees Celsius. This intense heat burns off all the organic material, the proteins, fats, and carbs, leaving behind only the non-combustible inorganic minerals. What's left is the ash, which is then weighed. So we've got moisture, protein, fat, and ash. What's missing? Carbohydrates. This is often the trickiest. The most common way to list carbs on a label is by difference. Scientists simply take 100% and subtract the measured percentages of moisture, protein, fat, and ash. Whatever is left over is assumed to be carbohydrates. It's an estimate, but it works well for labeling. For a more precise breakdown, especially for sugars and fiber, they use methods like high-performance liquid chromatography, or HPLC. This sophisticated machine separates the different types of carbs in a liquid sample, allowing scientists to identify and quantify specific sugars like fructose or glucose, and even different types of fiber. Finally, how do all these grams turn into calories? This is where the Atwater system comes in. It's a standardized set of conversion factors. For every gram of protein, we count for calories. For every gram of carbohydrate, it's also for calories. And for fat, which is more energy dense, we count 9 calories per gram. So you just multiply the grams of each macronutrient by its Atwater factor and add them all up to get the total calorie count you see on the label. So the next time you look at a nutrition facts panel, you'll know it's not just a bunch of numbers. It's the result of meticulous lab work, from oven drying and acid digestion to solvent extraction and high-tech chromatography. It's a fascinating blend of chemistry and precision that helps us understand exactly what's in our food. If you are a quality professional, subscribe to our channel. We have several videos with different topics that will make a total difference in your career upgrade.